Hello everyone, this is K. Ariaran from first MSc Microbiology Sacred College. The topic is Moraxella catarrhalis. Now we are going to see the general characteristics of the Moraxella catarrhalis. First is gram classification. Moraxella catarrhalis is gram-negative bacteria. Shape of the bacteria is cocci and are arranged in the diplococci. The motility of the bacteria is non-motile. Size is vary from 0.5 to 1 micrometer in diameter. The family of the Moraxella catarrhalis is Moraxellaceae, and the capsule of bacteria is present and endospores are absent. The respiration mode of the Moraxella catarrhalis is faculty anaerobic. Faculty anaerobic is nothing but it can grow well in both presence of oxygen and also the absence of oxygen. The optimum temperature of the bacteria is 37 degrees Celsius and optimum pH is 7 to 7.5. Habitat of the Moraxella catarrhalis is in the human respiratory tract at mainly nasopharynx. And this bacteria of Moraxella catarrhalis is discovered by the Hermann Lehmann and Fritz Neumann in 1896. Now we are going to see the disease transmission of the Moraxella catarrhalis. The disease can be transmitted by the three ways: by inhaling the respiratory droplets of the infected person to person, or direct contact with the infected persons. The next is virulence factors. The virulence factors of the Moraxella catarrhalis is outer membrane proteins, lipo-oligosaccharides, IgD binding proteins, beta-lactamase production, pilus structures. The incubation period of Moraxella catarrhalis is 1 to 3 days. The next is pathogenesis. The bacteria Moraxella catarrhalis is adhering to the mucosal surface of the respiratory tract. Structure allow the bacterium to attach to the respiratory epithelial cells and establish the colonization. Outer membrane protein 10 mpps plays a role in adhesion, evasion of immune system and nutrient acquisition. IgD binding protein help the bacterium avoid the host immune response by binding and inhibiting the functions of immunoglobulin D. Immunoglobulin D is nothing but IgD. It enhances the bacterium resistance to antibiotics and host immune defenses. Production of beta-lactamase can hydrolyze the antibiotics and to avoid phagocytosis by immune cells, and finally produce the symptoms of infections. Next we are going to see the clinical diseases of Moraxella catarrhalis. The first disease is otitis media. Otitis media is nothing, it's one type of ear infection. Moraxella catarrhalis is a common pathogen in otitis media, especially in children. It can cause middle ear infection leading to the symptoms such as ear pain, fluid in ear and hearing difficulties. The next infection is sinusitis. Moraxella catarrhalis is an implicated in cause of bacterial sinusitis. Inflammation of the sinuses. Sinuses is the region which is located side to the nose and below to the eye in the face that can result in the symptoms such as facial pain, nasal congestion and headaches. The next disease is exacerbation of chronic obstructive pulmonary disease. It is also shortly known as COPD. Moraxella catarrhalis is a known cause of exacerbation in individuals with chronic obstructive pulmonary disease. Infection can worsen respiratory symptoms, leading to increased cough, shortness of breath, and sputum production. The next disease is pneumonia. While less common than other respiratory infection, Moraxella catarrhalis has been associated with pneumonia, particularly in individuals with underlying health conditions or compromised immune systems. And the next disease is bronchitis. Moraxella catarrhalis can contribute to the acute bronchitis, characterized by the inflammation of the bronchial tube. This can lead to the symptoms such as cough, chest discomfort, and the production of mucus. The next disease is bacteremia and systematic infection. In individuals with weakened immune systems, Moraxella catarrhalis may cause more severe infection, including bacteremia. Bacteremia is nothing but presence of bacteria in the bloodstream and systematic infection. This is relatively uncommon but can be serious. The next is laboratory diagnosis of Moraxella catarrhalis. In laboratory diagnosis, microscopic examination, the gram staining of the Moraxella catarrhalis is observed as pink colored gram negative cocci bacteria and arranged in the form of chains. The motility test is observed as non-motile. In laboratory diagnosis, the second is colony morphology in culture medium. In blood agar, Moraxella catarrhalis is observed as a small grayish white color colonies. In mechanical agar, it may vary. Then in the laboratory diagnosis biochemical test, catalase test is positive for the Moraxella catalysis, oxidase test is positive, urease test is negative, indole test is negative, methyl red test is negative, Voges protease test is negative, citrate utilization test is positive, starch hydrolysis is negative, nitrate reduction test is negative. The next is treatment and prevention of the Moraxella catalysis. Beta lactam antibiotics is the drug of choice for the Moraxella catalysis. Macrolides, fluoroquinolines, trimethoprim, sulfomethoxazole are also be used for the Moraxella catalysis. And hygiene, respiratory hygiene, avoiding close contact with the infected person can be prevent from the Moraxella catalysis infection. And also the vaccines are available for the Moraxella catalysis infections. Also, the clear notes of the Moraxella catalysis is uploaded in our blog. The link of the blog is attached in the description of this video. Thank you.